Do you see everyone is okay with the lecture?
I think so.
Okay. So, good morning, everyone. So, yeah, it's. I actually had some fun yesterday. I thought the part where we looked at the videos yesterday, that worked quite well. It was not too stressful, and actually we finished a little bit early in our group. And so today I will tell you a little bit about general vascular development. So how during organogenesis, how do blood vessels develop? How do they make a network? And I will particular talk about three subjects that were touched yesterday during these video analyses. So you will hear this thing again, and I will give you some more information about these issues that we addressed yesterday. So vascular development, that means it's development of an organ. It's the first organ to develop in a vertebrate embryo. It's an organ that is made up of tubes. It's not the only organ that is made up of tubes. The lung, the kidney, mammary glands, the gut, they're all tubes, but very particular what the vascular system is, it's actually a network. So it's not tube that are blind-ended or single tube with one opening and one, or with two openings rather. It is a network, and this interconnection is very important to efficiently transport the blood through all the body, you know. Evolutionary speaking, blood vessels are an old invention. It's not new. We have the most simple metazones are the cnidarians, which you see on the top. So they are these diploblasts. They don't have a mesoderm, and they don't have any vessels. And then you go to these arthropods like Drosophila, locust here shown on the top right. They have a heart, as Marcus already mentioned yesterday, but then it's an open tubular. It's an open system, so the blood is just flowing through the body, but it's not really channeled within blood vessels. So, but on the other hand, we see closed vascular systems already in annelids, and then of course in all the vertebrates. And the role of blood vessels is multifold. It's not just bringing oxygen, nutrients, but it also carries away waste and also transports hormones. And actually, it's the interface between our organs and the outside world. A few interesting facts. So as I mentioned before, it's the first organ to develop in a vertebrate embryo. It's the first functional organ. And that is because the embryo already needs it. It doesn't need a kidney in the beginning. It doesn't need a brain. The brain is for later times. It has to develop, but actually this cardiovascular system has to work for the embryo to develop. And that makes sense because it's growing. It has to fulfill most of its functions already during embryogenesis, whether it's a mammal or a fish. Especially mammals, it's important because the embryo sits in the placenta, and there has to be this exchange of nutrients between mother and embryo. Yeah, so it's the interface between the outside world, because it brings stuff from the outside into the cells, basically. And very particular is that there's a single cell type that is associated with blood vessels, and these are the endothelial cells. So it's a very special cell type. You will hear more about this later. I will discuss it. These cells are the inner lining of the blood vessels. So blood vessels can have many layers, but the inner layer, so the lining of the tube, is the endothelial cells. So capillaries are basically made of endothelial cells. There is 1,000 million, billion, a trillion. So in American counting, it's a trillion endothelial cells that line our blood vessels in the body, and that's a huge amount of surface covering the adult. And the heart pumps about 14 to 15 liters, 14 to 15,000 liters of blood every day. So it's a very, very good pump. It's very reliable, and we have to take care of it throughout our life because it's really doing, as Donald Trump would say, it's really doing a great job. Okay, and this is how the system works. The blood vessels are, you see that on the right, so they are lined by endothelial cells, and they're surrounded by the basal lamina, and then you have supporting cells. It can be smooth muscle cells, it can be pericytes. They're important for the maintenance, for the well-being of the endothelial cells. They also can control blood pressure because they can produce hormones that can control the contraction of the vessel. So, or actually the smooth muscle cells are, they can increase or decrease the diameter of the vessel depending on whether you have adrenaline or not. So if you get scared, you may get a contraction of your smooth muscles, and that will really increase your blood pressure. Okay, the core of the system, of course, is the heart, and in mammals, we have the luxury of having a 4-chambered heart and 2 circulatory loops. There is one loop that you see on the sides that goes through the lungs. Okay, so you have the blood coming from the vein, and here, and then from the heart, it can go through the lungs, so through the right heart chamber. And when it goes through the lungs, it can pick up the oxygen, and then it ends up in the left heart chamber, and then this oxygen-enriched blood can go through our body, through the capillaries, to all our organs, to the liver, to the brain, whatnot. And then it can supply the oxygen, pick up the CO2, and then it goes back to the heart and to the lungs and so on. In fish, the system is a little more simple. And you have a system where the second loop is left out. There's some evolutionary interesting facts about fish and the swim bladder being, the ancient lung. I'm not going into that, but there is already some evidence that also in fish, in the swim bladder, the blood vessel can also pick up oxygen. That's an interesting hypothesis that was put up about 40 years ago, 50 years ago. It is interesting. If you ever happen to come across this the subject, you may want to read it. But for the most part, in the fish, it's a bit more simple. You have a 2-chambered heart. Actually, the first thing here on the right is called, that's the atrium, and then you have the ventricle. Then the blood goes through the gills. So these are the gills in front of that I showed you yesterday. The gills are the one that do the gas exchange in the back of the head, and then it goes through the body, and then back to the heart. So it is a bit less complicated. Every organ has their own type of vessel, and that requires...
Henry?
Yes?
Can you go back one slide?
Yeah.
I find this a bit funny. I find this drawing a bit funny because on the left side, also you have the heart in the fish, then it goes through to the left, up the gills, gets oxygenated, and then the drawing includes going back to the gills to get deoxygenated.
No, these are the capillaries in the body.
Yes,
These are the capillaries in the gills. Yeah, it's not a good drawing.
Okay.
I promise to, and I'm not promising. I'm too lazy to change that. Yeah, it's not a very nice drawing.
Yeah,
I make one myself, maybe.
Mm-hmm.
It's always also for the students. If you ever happen to write your master's thesis and PhD thesis, make your own drawings. You know, you don't do yourself a favor by copy pasting everything. Anyway, okay.
No, it is surprising how many figures out in the literature are conceptually wrong. And this.
Well, this is correct. It's just drawn awkwardly because they draw the gills and the capillaries in the same way. Yeah. So.
Okay.
So it's not incorrect. It's just not very well done. Yeah. Okay. So different organs have different functions, and the vascular system is very adaptive. It's adaptive in a way that it can change in situations, but it's also adaptive in a way that different organs provide blood vessels with different qualities. And that means that these endothelial cells can look vastly different. They have a lot of different phenotypes. And that is just shown in this drawing. I don't have to go through all the details here, but you can appreciate that while here in A, this is what vessels look like in capillaries. You have a single lining of these endothelial cells. So this would be the lumen here on the top. And here you have the basal lamina. You can have these little, like vacuoles. They can so that macrophages, leukocytes, they can go actually through these endothelial cells and migrate, and they can go out from the blood vessels, go into the tissue, and attack, sort of an invader, like a pathogen, and kill a bacterium, just like that. They can also migrate between the endothelial cells. It's a different subject. There can actually be holes throughout these endothelial cells. So you have then pores with a diaphragm, and you can also have all these different clefts and so on. Because, for example, in the liver, in the bone marrow, especially in the liver, there has to be a lot of transport from outside from the tissue into the blood vessels. So you want to facilitate that cells can go in and out, that particles can go in and out, like lipid droplets and stuff like that. So blood vessels are extremely diverse, and the diversity is based on the different phenotypes that endothelial cells can acquire. So they're very plastic. And in the lab, we study angiogenesis and endothelial cells in different model organisms or model systems. Of course, you can do in vitro studies, as shown here. Then you have cell culture. You can take primary cell cultures. You can take primary cell cultures from different organs. You can take brain endothelial cells, lung endothelial cells. That's actually a subject that is important right now. People are looking at what coronavirus is doing actually to lung endothelial cells, because it has been shown that particularly coronavirus causes inflammation of the capillaries in the lung. And that is part of the pathology of coronavirus, is that it causes an inflammation in not just in general the lung, but in the microvasculature of the lung. And so people are particularly now looking for lung endothelial cells. So where do they get them from? They get them actually from people that had lung cancer. So from the biopsies, and then you try to extract those lung endothelial cells and put them into culture. So you can do primary cultures. You can also make cell lines that are endothelial cells. You can use embryoid bodies. You use embryonic stem cells that you can differentiate, and so on. You can do a lot of things there. But as developmental biologists, and we are particularly interested in using sort of in vivo models, whole organisms, and so you can use the mouse. The mouse has also, the same basically blood vessels as we have as humans, and you can use different organs where you want to study. and very popular because of its very nice morphology and accessibility. People use the blood vessels that are in the retina. So you can take out the whole retina, and it is not just has neurons, but it also has blood vessels, and that is shown here. So you have here these arteries, and you form this plexus, this network. You see that there's a hierarchy in the structure. You have big vessels, you have small vessels, and so on. You're by now familiar with the zebrafish, and also the quail is kind of interesting because you can look very nicely at cell dynamics. Okay, and as I pointed out many times before, different model systems serve different purposes and different experimental designs. So going to the fish more now. So we look here at the basic vascular anatomy of a fish that is about 3 days old. It's expressing a transgene. This is what we call FLIC1 GFP. It's not flea, but it's FLIC. It doesn't matter at this point what it is. Actually, it's a VEGF receptor 2 gene that is regulating here EGFP expression. And on the right-hand side, you see what we looked at mostly in this course. We have the trunk vasculature. We have the different blood vessels that are pointed out at the bottom. So we have the dorsal aorta that flows from the left to the right. We have the posterior cardinal vein here that brings the blood back to the heart from the right to the left. Blood cells can go from the dorsal aorta up to this vessel up here, which is called the DLAV, dorsal longitudinal anastomotic vessel. Anastomosis means things coming together. It's sort of a fusion. It's a vessel that's basically connecting. It's an interconnection between the artery and the vein. And then from the segmental vein, you go back to the heart, and that we saw yesterday in a video how that is happening, how these circulatory loops are made. On the left, you see the brain. The matter is much more complex. So it's very difficult to point out a single particular vessel here. It's a bit more complex, so I leave that out for the moment and for today. And then, of course, because of the transparency, and that's what we showed you yesterday in these videos in the morning, you can just take a light microscope. You can take even just your iPhone, I guess, and if you have enough zoom in there, you would be able to see the blood flow in the embryo. And just as I described before, now you can see it actually live. You see the blood flow up and down in the segmental arteries and veins, and you see the pulsation in the dorsal aorta, and less pulsatile flow in the vein because there's a lot less pressure. What you also see here very clearly now on the left is that these veins, that these vessels, these intersegmental vessels, are sort of squeezed in between the somites. And that is an interesting observation later on. You see that that's not, you can't take that for granted. You could just say maybe, oh, okay, that's where the space is. The somites constitute a mass of cells, and this is something where blood vessels cannot go because simply they block the way, and they just take the easy way and use the cleft, which is called the intersomitic fissure here, to move up. Actually, what the question is, what makes this vessel go up and sprout in this direction? This is something we also talk about today. Okay, now I think most of you have a good understanding of this, these tools that we make. So what it is to make it transgenic, I just make it very brief. So when you start working on a system, let's say you go back here and you see this pattern and you say, okay, I want to study the cell biology of this. I want to know what these endothelial cells are doing. You want to make a transgenic line that marks these cells. And in the early 2000s, there was no line available to mark these cells. There was no transgenic. So the first transgenic came in 2002. And so Nathan Lawson, a postdoc in the time, what he did was he cloned a gene that is expressed in endothelial cells. So he did whole mount in situ hybridization, and he found this particular gene. FLI1A is expressed specifically in endothelial cells. So this is a whole mount in situ, and you see there's these two rows, these bilateral rows of cells. So there would be here the midline here of the embryo, and with the neural tube and the nodal cord, and you see on the sides here, you see in the lateral mesoderm, you see this group of cells expressing FLI1 coming together in a later stage embryo, and then in this stage, you see there's a dorsal aorta and the posterior canal vein, they express FLI1. Okay, so he cloned the genomic region, which was, I think, pretty big, like 15 kb, and inserted EGFP in that, just regular cloning. And he injected and he cloned EGFP right next to the endogenous ATG of the FLI1 gene, and injected that into the embryo. This is a drop of about 2 to 4 nanoliters, so just so you know. And then you look for transient expression of this GFP in your embryo. You raise the fish, you can incross them. You see whether any of them now in the F1 generation have nice expression. You can also confirm, if you don't look for expression, you can do PCR analysis, whether the genome of these fish have picked up your insert, your plasmid, and then you raise the F1, and then you go ahead. And if you're lucky, you find about 1 transgenic carrier from the G0 here out of 20 that are transgenic. So ideally, you want like 4 independent insertions out of 100 or 200 fish, and then select for the best expressing insertion. And that's the line that you keep. You better keep 2 lines to be on the safe side. But this is how we do it in the lab. Okay, so now we have a marker for these endothelial cells. We have a FLI EGFP transgenic. And then one thing we can do, for example, is to see actually where do endothelial cells originate? When do they emerge? When do they first express? Do they emerge in one place and then expand? Do they emerge in different places? And you can do that by doing a time lapse. So I'm showing you a time lapse now of the embryo of myxomitogenesis, and you see that there is several populations here of cells. You see emerging here all along the body axis. Actually, what you see here is an independent group of cells, and some of these are actually not endothelial cells, but they are blood cells. So FLI1 is also expressed in early blood cells. So that's why you had this mass of cells also inside here. And then you also see the sprouting is already emerging from the dorsal aorta, and the first sprouts are forming here in the front. Oops. So how are blood vessels made? So we have endothelial cells, we know where they come from, and the question now is, how do they make a vessel? Because the first endothelial cells are called actually angioblasts, because they're not part of the vessels, they are not fully differentiated. And they emerge in the lateral plate mesoderm, as you saw quite early during somatogenesis. And they are sitting in 2 rows on each side of the midline. And now they have to find each other and then actually building a tube out of being just a group of cells. Right? It's like Lego pieces, and then they have to form a tubular structure. And this is what you can see here is actually the first vessels that are made are made in a process called vasculogenesis. So this is the de novo tube formation from just cells that are hanging out, migrating towards each other, coalescing, and then making a tube. And this migration is that you see here is that cells are coming together and then forming a rod. And question, is this a tube yet? I wouldn't think so. But this is basically the first step in blood vessel formation. Vasculogenesis is that you have groups of cells that are coming together, they're merging, and then form a cluster of cells when they aggregate. Then the lumen is formed in a certain way, and I just show you the schematic on the right-hand side. Is that the cells come together from both sides in the midline. They generate a contact. This contact is expanded, and in the middle, you have a membrane domain that becomes a bit specialized, and that is going to be the luminal membrane, and we call that also the apical membrane. So what you have here is a cell that has uniform membrane surface all around. Then they can make a junction, and within this area, you insert membrane material, and that membrane material is special. It has certain proteins sitting on it, and it's anti-adhesive, for example. They express a protein that is sort of like a Teflon. It's an anti-stick. It's an electrostatically negative protein, so the membranes don't stick to each other. And this membrane here is the apical membrane, so that is the luminal side of the vessel, and the luminal side basically of these endothelial cells having an apical and a basal side. And they fulfill different functions. So the basal side is important to make attachment to the outside world with the basal lamina, with the extracellular matrix, and the inner side, the apical membrane, is important for communication with what's going on inside. That's where you measure blood pressure, blood flow, and so on. This is showing again as a summary in a schematic. This is a cross-section through the embryo at somatogenesis, and then to 1 day and 1.5 day embryos. You start out with these endothelial cells that have been specified in gastrulation. Remember yesterday we talked about gastrulation and dorsoventral patterning. So these cells have a ventral fate. So this is ventral mesoderm, lateral plate mesoderm. They have relatively high levels of BMP in the beginning. They migrate over the endoderm to the midline next to the notochord here. There they segregate into a dorsal and a ventral population, and then they have formed the dorsal aorta and later on the posterior canal vein. How exactly this is going on, there has been some debate, but I think in a nutshell, this is all that you need to know, is that the earliest vessels are formed by these angioblasts early on. They migrate in the middle, they aggregate, and they form two separate tubes. One peculiar subject was always what makes these cells move inside. So why do they do that? Is that is there something here in the endoderm that makes them migrate to the middle, or is it something from the notochord? So what we all know in notochord important signal from the notochord is head shock. So people thought head shock is important, a signal to be an attractive signal for these cells to go to the middle. Another molecule that we talk about in a lot of detail later on is called vascular endothelial growth factor. People took it for granted actually because it's such an important molecule that it would be somehow there would be VEGF here and the cells would migrate towards this vascular endothelial growth factor source somewhere around here. Actually, that was wrong, and it took from this paper here, from this knowledge here from 2002, it took almost another 15 years to actually find the signal that makes this angioblast go to the midline. And so this migration was done by a particular pair of molecules, apelin APJ signaling pair. And this is just for you. You don't have to understand apelin APJ signaling, but just to show you how we do this kind of analysis. So at the top, you have this wild type situation where you see this midline migration of this angioblast very effectively, very fast. We don't have a timestamp in this movie, but this is a matter of a few hours that these cells aggregate and actually form dorsal aorta. If you lack apelin AB, so that's a receptor, the cells just stick there around. So that's a very impressive phenotype. This is something that we could do in the practical part next year also. They're not doing anything, so the signal is completely gone. Okay, now this could have been a task. The next slide shows you the task we actually had yesterday, and that was
Henry?
looking at angiogen. Yes?
So apelin is a receptor?
Yeah, I'm not sure. I thought so. I'm not sure actually at this moment. I just you caught me on the wrong foot now.
Okay, so I'll tell you. So there is a receptor expressed in these cells here. So can you show the cells?
You have to. I can't see.
Yes,
you.
I know. On the upper slide. So the apelin receptor. Apelin is a ligand. Apelin receptor is expressed in endothelial cells.
Okay.
As far as I know, and the ligand is actually expressed in the midline. So the midline attracts.
But it was not VEGF. It was not head shock. Yeah.
Exactly. So there is a ligand, but this ligand is apelin. As far as I know, and apelin receptor.
APJ. Okay.
Yes, is expressed in the endothelial cells, and then they migrate towards the source. So the concepts were right, but the molecular things were kind of different. Yes.
So the basic point is that you have receptors and ligands expressed in different cells, and if it's a cytokine, the cells that have the receptor will go towards the signal. That's a basic concept, and you need both. If you miss either one of them, the receptor or the ligand, this migration will not happen. Agreed?
Yes. I didn't want to correct you. I just wanted to.
No, but I did.
I thought you would add it anyhow, and then you went to the next slide. And I think it's a pity for the students not to see. There is a source, and cells migrate towards this source.
Exactly. So, okay. So task number 1 yesterday was trying to understand what's going on in terms of morphogenesis. What are the patterns of development during ISV formation? And we discussed in detail these movies, and I don't have to go into too much detail anymore because we did that yesterday already. We saw these cells from dorsal aorta moving dorsally, forming these structures here, which is the DLAV. We saw that there is lumen formation, because the lumen, you see that as it doesn't have GFP because the lumen sort of pushes the cells to the side, and then the GFP goes to the side And that's how you sort of indirectly visualize the lumen, by the lack of GFP expression. So we see this here on the right-hand side conceptualized a little bit. We have these sprouts coming out of the dorsal aorta. The tip cells here, so the first cell we call the tip cell, they sort of fan out to the left and to the right. We don't exactly know why they do it just like that, whether there's a particular process that regulates that, we don't know that. But nevertheless, they interconnect with each other, the cells. So what you have is not cell fusion, but you have vessel fusion. So when you say what the cells do is they fuse, it's not correct, they connect. But then on the vascular level, on the vessel level, they do actually fuse. But this is a dead end, and you would have different branches here with having the same pressure. There would be no efficient blood flow from here to here. So the system works in a way that you have sprouts, secondary sprouts. They come from the PCV, the posterior canal vein, or from the caudal vein, and you see these sprouts coming up here. And you see another one, I think, somewhere here. And they hijack these arteries and transform them into veins. And you saw in this other movie that I don't have here in my presentations, is that the formation of a vascular loop is then efficiently made, and you actually have then circulation from the dorsal aorta through the segmental artery, DLAV, back through the segmental vein, and to the posterior cardinal vein. Okay, so that was the wild type, and then the next step after the sprouting is this anastomosis. I just want to show this in a little bit more detail so you get more of a feel for it. So the situation we're in here now is we have two sprouts, we have two tip cells, one on the right side and on the left side. They have reached the dorsal end of the embryo, so that's the level of the spinal cord here. They interact, they touch. There's more touching going on. Then you see this lumen formation, so the lumen is creeping up through the cells or between cells. You can't see that here, but then eventually there's lumens, and the lumens interconnect, and when they interconnect, they allow blood flow. Okay. This is again shown in the next slide. I don't have to discuss this. This is for you, for your bookkeeping, so that you have an easy to look at schematic if it ever occurs to you that you want to understand. So then you don't have to look at the videos, because your handouts don't have videos. So this is sort of the essence of what we've seen in the videos. Then we go to task number 2, called sticky business. So here we looked at the role of a particular adhesion molecule, V-equaterin, and we can look at the role of V-equaterin by making a knockout. And then when we analyze the knockout and compare that to the wild type, we get an idea of what is the requirement of this protein in blood vessel formation. And there should have been a different slide, but nevertheless. So we look again at this wild type embryo. We're all familiar with this, but every video looks a little different, depending on stages and levels of expression and whatnot. So there's always a little bit different things to see. There's some variation in the process. But as you saw yesterday, what we saw was there was this general sprouting. There was some funny things. The DLAV was not forming properly, but most important, you saw this breaking up of these sprouts ventral to the tip cell. So there was a disconnection between what we call the stalk and the tip cell. And that is not so surprising, because we say it's an adhesion molecule. So it is important to maintain stability during the sprouting process. This could have several reasons. It could be that generally adhesion between cells is anyway downregulated, and then you lose V-caterin, the whole system becomes more unstable. Or it could be that the extreme pulling forces from the tip cell on the stalk cells, and if you don't have V-caterin, this connection breaks. Okay, so that is no big deal, but it's consistent with this protein having a role in adhesion. Okay, so this is what V-caterin looks like. It's an adhesion molecule. It sits in the membrane. It can form dimers in cis, so on the same cell, or in trans between cells. And then it forms clusters, cis and trans clusters. And it has a transmembrane domain. It's a single pass. And then in the inside, it has a lot of, and this is only a few shown here, it has a lot of protein-protein interactions. And most importantly, it is connected to the F-actin cytoskeleton via other proteins. It's connecting the junction to the cortical actin cytoskeleton. So it's important for actually F-actin mechanics of the cells, which we won't go into today. But it's also important for signaling, because you know that beta-catenin is a signaling molecule. Actually, it's a funny protein because it's important for structures. It is important as a structural protein at cell junctions by binding to caterins and making the interconnection between caterins and F-actin. But it's also known to be an important signaling molecule in canonical Wnt signaling, and I'm sure you've heard about that already. So that also means that a lot of V-caterin can sequester beta-catenin to the junctions, and thereby, by playing with the levels of beta-catenin here, it can influence Wnt signaling in the cell. So it's also important for signaling. And there's a lot more about this molecule. I'm not going into this. So it's important for stability and during sprouting angiogenesis, but it's an adhesion molecule. It is vascular specific. So there's a lot of caterins expressed in the embryo, in the vertebrate, and they have more or less tissue specific expression, and vascular endothelial caterin, as the name says, is specifically expressed in endothelial cells. So this is a way that endothelial cells can make specific connections between them. So it can be used not just for adhesion, but also for recognition between cells. So that makes you think then, what happens in anastomosis is that cells, tip cells, they go up, they want to form the DLAV, right? And they have to recognize each other. So we looked in particular with V-caterin mutants, what happens when endothelial cells don't have V-caterin? Can they still recognize each other? Is it not just adhesion, but also cell-cell recognition? And so these are a couple of movies where we use two different markers, F-actin and a cell membrane marker, to better see the dynamics of the filopodia, because the connections are initially made by the filopodia, as you see in this video. Tip cells are rich in filopodia. They probe the environment for signals and for other cells, and then you see these filopodia they touch, and then this filopodial contact is then transformed very quickly in a permanent contact. There's some touching going on and detaching, and then they actually make this contact. So they have touched each other, they've recognized each other, they say, okay, we both have, all what it takes to make a contact, and it's a specific contact, and then they can initiate vessel fusion. So if you don't have V-caterin that's shown at the bottom, the filopodia take a long time. They seem to touch, there's interactions, but it takes a long time for them to get together. So in one hand, of course, yeah, the question is answered. V-caterin is important for this recognition, but it doesn't seem to be absolutely essential because the contact in the end is still made. I mean, there's a lot of defects here that I can't go into, but in principle, the contacts are made, although it's slowed down. So it's attenuation rather than inhibition. So then we thought, okay, there must be more to this. And okay, V-caterin is not the only junctional molecule in these endothelial cell junctions. There's more. There's claudins, they're important for tight junctions, and JAMs, they are also they're junctional adhesion molecules. One of them is ESAM, and we have V-caterin and others. So there could be other molecules contributing to anastomosis and specifically to cell recognition. And in that situation, you have to make an educated guess. Let's say there is several claudins, there is 3 or 4 JAMs, there's ESAM, there's nectins, there's several nectins, they're all expressed in endothelial cells. But the question is, which ones of them have adhesive power and which of ones are really endothelial specific? And so by talking to some experts in the field, we decided that we would look at this molecule. It's called ESAM. It belongs to the JAM family, and it is called endothelial selective adhesion molecule. It's really quite specific for endothelial cells. There's some other cells too that express this, but very few. So it's really an endothelial specific gene. It has a lot of adhesive power. What we talk about here is 1.5 nanonewton per molecule or something like that. And then, okay, we made a knockout. And then we checked what happens when we knock out this molecule here. That's wild type again, that's boring. And on the bottom you see, well, that looks like wild type. That's also boring. So this molecule doesn't have a function. So maybe that's disappointed, no phenotype. It seems to be a non-essential gene. But then we said, what happens if we do both? Now we had a mild phenotype with the V-caterin, and then we make a double mutant. And then you see again V-caterin at the top. That's the phenotypes you're used to. You know, we talked about this. These cells going down. This looks a bit weird. We still have these disconnections, but you don't see that here in this particular video because it's only 30% of the vessels. So once in a while you hit a video where you don't have these disconnections. And then in the double mutant, what happens suddenly, basically these vessels completely fall apart. So if you lack V-caterin and ESAM, there is no, once the cells start to sprout, they're not able to reinitiate any blood vessel morphogenesis, it seems. And then particularly if you look at anastomosis in the double mutant, these filopodia, they don't make any real contacts anymore. It's just cell migration. So cells start moving over each other. And basically, they don't recognize each other as endothelial cells anymore. And that means that V-caterin and ESAM together are the key molecules that are essential for endothelial cell adhesion and recognition during morphogenesis. Okay, task number 3. Actually, I'm running a little bit late. Anyway, so I tried to speed up a little bit, and that was analysis of endothelial cell migration and proliferation Notch mutants. And I can't talk about Notch without talking about vascular endothelial growth factor, VEGF. And so VEGF was discovered about, I don't know, 25 years ago. And there was a very conceptual review 20 years ago by this guy, Werner Riesau. And make a long story short, on the right-hand side, you see basically all the processes that are considered to be important in blood vessel development. You have the induction of the mesoderm. You have, excuse me, just 1 second. Okay, yeah, so these processes that go on, and just look at how often this word VEGF receptor or is here. So it's involved in everything. Okay, so it seems to be the most important factor in anything that happens in vascular development. Okay, that's all from this one here. It comes in different flavors, different isoforms, different receptors. It's a very complex system. You don't have to know this. What you have to know is that VEGF-A is the key molecule that actually activates endothelial cells to sprout, particularly when it comes to these intersegmental vessels. Okay, so in this particular situation where we have this situation of dorsal aorta and ISV sprouting, VEGF-A is the key cytokine that activates these cells. And if you look at this situation in G, without VEGF receptor 2 signaling, this is an inhibitor for VEGF, you do not have any sprouting. Okay. So that answers the question is what makes the endothelial cells sprout? It's actually an activation via VEGF-A signaling through VEGF receptor 2, which is in the endothelial cells. Okay, we won't make a break. I show you a video now about the relevance of angiogenesis in tumor biology. And I have to do a new share here. How does this work? Hold on a second. Okay. See, this is difficult to do in home office. Okay.
For today's cutaneous wound healing lecture, just remember that during wound healing, first there was formation of blood clot and hemostasis. Then,
okay, sorry.
Mammalian cells obtain the oxygen and nutrients they require to survive from nearby blood vessels. Because of this, cells are usually located no further than 0.1 to 0.2 millimeters from a capillary. Tumor cells also require a blood supply to grow. Without this, solid tumor growth is limited to about 2 millimeters, or approximately 100 to 300 cells. Small tumors without blood vessels may lie dormant for months or even years. However, tumors can develop their own blood supply by a process known as angiogenesis. The process begins when tumor cells secrete angiogenic growth factors that stimulate endothelial cells lining nearby vessels. In addition, some tumors also require activation of certain growth factor receptors, such as epidermal growth factor receptor and RET, in order to grow and spread throughout the body. Therefore, agents that block angiogenesis and tumor cell growth have the potential to be more effective anti-cancer therapies than agents that act on only one of these pathways. Of the angiogenic factors produced by the tumor, the most important is thought to be the vascular endothelial growth factor, or VEGF. VEGF binds to specific receptors located on the endothelial cells lining nearby blood vessels. VEGF binds to the extracellular portion of the VEGF receptor, promoting receptor dimerization. This activates the receptor's intracellular tyrosine kinase domain, and autophosphorylation takes place. This allows specific intracellular molecules to recognize and dock onto the VEGF receptor, propagating a signal from the cell surface into the cell. VEGF signaling within the endothelial cells stimulates the formation of new vessels that grow towards the tumor. The tiny tumor now has a supply of oxygen and nutrients, allowing it to grow and to metastasize to other parts of the body. Activation of certain growth factor receptors is implicated in tumor cell growth. For example, the epidermal growth factor receptor, EGFR, is important in many cancers, and RET is important in certain types of thyroid cancers. Growth factors bind to the extracellular portion of their receptors, promoting receptor dimerization. This leads to activation of the receptor's intracellular tyrosine kinase domain and autophosphorylation, which in turn stimulates a cascade of downstream signaling events. The consequences of receptor signaling are tumor growth and metastasis. AstraZeneca is investigating orally bioavailable small molecules that target the intracellular tyrosine kinase domains of the VEGF receptor, EGFR, and RET kinase. These studies are evaluating the effects of inhibiting ATP binding within the tyrosine kinase domain of the VEGF receptor to prevent the signal transduction that would otherwise lead to new tumor blood vessel formation. The effect of inhibiting signal transduction via EGFR and RET kinases on tumor growth and survival is also being evaluated. In the absence of a blood supply and inhibition of growth factor signaling, the tumor remains in a dormant state. In addition, the ability of the tumor to metastasize is reduced.
Henry? Henri?
Yeah.
Okay, now I hear you.
Yeah. So,
maybe some people didn't see the movie. In the chat, somebody said that the movie was stalling all the time. So
okay, but they have the link.
Yes.
So the students have the link, so they can just. Yeah, maybe that's a bit different. Also, my computer was starting to heat up here. It's a hot movie. Nevertheless, so I have not much time left. Okay, so we were talking about drug discovery, how people try to develop drugs that prevent tumor angiogenesis, and actually the zebrafish is also a great system to look for drugs, and you can use different assays that you can use in order to test your substances in sort of a 96-well format. And so instead of doing this traditional way of identifying a target and going through all these different steps of compound identification and structure-based design, which takes up to 15, 20 years, another route one could go is to do a zebrafish screen, high throughput, right on embryos with those different assays that we have in the zebrafish, testing the best compounds, and then put them from this in vivo setting relatively quickly into mammalian models and to human trials. That would save about 5 years. And actually this way, a drug that is now involved in blood bone marrow transplantation was actually discovered by this approach, that you first test your candidates in zebrafish and then take the best hits and develop those further. I'm gonna skip this part now because I want to basically discuss this video now. So what I wanted to tell you in these slides before is that VEGF, okay, I go one slide back, and that is shown here on the right-hand side, that this molecule VEGF that you saw the video, and that is this important chemokine here in ISV sprouting, is actually a regulator of notch signaling, and that you have a tip cell experiencing high VEGF signal expresses this ligand delta-like 4, which is a ligand for notch, and that this regulation leads to a differential behavior of tip cell versus stalk cells. But in the end, what this is important for to know is that you need VEGF to sprout, but that this VEGF at the same time also regulates notch signaling, okay? And in an indirect fashion, that the tip cell then signals to notch in the other cells. That's all that is important here, that you have a primary signal leading to a secondary effect in a different signaling pathway. And this is the notch pathway, and that's what we looked at yesterday in these videos. And that was shown here, looking at these proliferation patterns in the wild-type embryo. And then we see there's only 3 cells per ISV, while when we inhibit notch signaling, we see a lot more cells. Okay. Now, the question is, are there more cells because there's more proliferation, or are there more cells because there's more migration? And then we saw in the wild type that usually there is 2 cells entering the sprout, and each cell in this video divides. Only the tip cell divides, but we know actually from other movies, this is a small sample size here, that also the tip cells and also the stalk cells usually divide. But in the end here, in these 2 instances, we have the tip cell that divided once and the stalk cell migrated in and didn't divide. So you have 3 cells. I could show you other videos where the situation is a little bit different. If we inhibit notch, it becomes obvious there's a lot more cell migration here. So the cells, there are more cells that are dividing, but every cell divides once. There's one cell number 3-1, does a second round of division, I guess. But there is contribution of both. You have more proliferation and more migration. So it's a single pathway that seems to regulate both migration and proliferation of these endothelial cells. And what is important to know is that when you think about it, what is notch? What is the role then of notch here? Why is notch important? Well, it seems to be sort of a counterbalancing response. So what this looks like, if you have no notch, it looks like you have an overactivation of the VEGF pathway. So you have these cells overactivated. So it looks like, as in a first step, VEGF-A is activating endothelial cells. It induces them to migrate. It induces them to proliferate. And then, in a second step, you have Notch. And what Notch is doing, it is sort of antagonizing some of the effects of VEGF-A. It is reducing migration. It is reducing proliferation. And if you don't have Notch, you say the effect that you have actually then full-blown angiogenesis that is becoming deregulated. Okay, so it seems that in this case, Notch is downstream of VEGF-A, and it is a sort of a negative feedback. Okay. That is that. And then task number 4, and that was the last task, was a question of endothelial cell navigation, endothelial pathfinding, and we had this very interesting mutant called out of bounds. And if you look at these mutants at 2 days of development, 1.5 days, 2 days, you see, instead of having these segmented organization of these endothelial, of these intersegmental vessels, you end up with this disordered network. You have blood vessels everywhere. They don't follow a certain pattern anymore. And if you look at these videos, you see in the wild type, of course, this metameric organization, that is there's one vessel per segment, and the vessels go between the somites. Not so in the mutants, where they sprout originally mostly in the right place, but then they go all over the place. So highly disordered. So they seem to have lost orientation. And the question is then, of course, how does that happen? So it seems to be a pathfinding defect. There was one hypothesis put up yesterday that said, okay, maybe it's a molecule that is expressed in the dorsal aorta and that makes the cells migrate away from the dorsal aorta. And if they don't have that molecule, they hang out more closely to the dorsal aorta. That's one possibility. The story, although, is a little bit different. And what we have here now is, so we have a disorientation of these cells. These cells, they do go all the way up, but they go also all the way all over the place. And I want to give you, in the last few minutes, I go about 5 minutes over time. I hope that is okay for you. That people started thinking about how actually do find endothelial cells their way through the tissue that they make the proper connections. And it was already in the 1600s shown that actually the patterns of nerves and blood vessels, capillaries, no, they didn't know about capillaries at those days. Blood vessels and nerves have similar patterns of distribution. And if you look actually on the right-hand side, so these are now capillaries. This is in the skin. These are nerves in green and blood vessels in red in the skin of a mouse ear, I think, or anyway, mouse skin. And you see that they actually align very closely to each other. So especially where it's yellow, they actually, they seem to hug each other. And then the concept was that nerves and vessels may use the same molecular cues or serve each other as cues for direction. And in the end, it turned out actually that endothelial cells and growth cones of axons express the same molecules. And some of these signals are attractive signals, and some of these signals are repellent. So you have repulsion and attraction. And it turned out that in the case of plexin D1, or that the OBD mutant was a mutation in plexin D1. So there is a repulsive signal that is missing. So there's something repulsive that prevents these cells going all over the place. And interestingly, there is a ligand, it's called semaphorin. In this case, it's semaphorin 3A. It's expressed in the somites. And this wild-type interaction between plexin D1 expressed in the endothelial cells and the semaphorin expressed in the somites prevents these endothelial cells crossing these borders. So that means in the end, what I alluded to in the beginning of this lecture was maybe these somites are just physical barriers, but they also, it turns out now, they're not just physical barriers, they're actually molecular barriers. They are territory where endothelial cells are not allowed, and that is because of this repellent interaction between plexin D1 and semaphorin 3A. And this is the summary of these different molecules and how they work. You have repulsion cues, you have attractive cues. VEGF receptor 2 is an attractive cue. VEGF receptor 2 signaling leads to the activation of delta-like 4, which leads to activation of notch 1, of notch in neighboring cells. So that is regulating then the behavior of the stalk cells. On the other hand, plexin D1 is a repulsive cue. It prevents angiogenesis sprouting in those territories where plexin D1 becomes activated. And with that, I close my lecture. I'm open for questions, and if you have questions now, you can also ask them now. Markus, if you have some comments, or Daniel, if you have picked up some background noises.
No, I'm okay.
Okay, then I hope you all the best for...
Wait. Also, you can make your final statement, but don't turn down the camera because we have these blockers all picture now.
Okay, yeah. So I wish you all the best for the rest of your block course. Good luck with this afternoon, that Martin can answer all the questions, and for your presentations. I may listen in on a couple of them, of these presentations, where blood vessels are involved. And yeah, don't suffer too much from Corona. This is in the cartoon, you see something that I've been going through a little bit over the last couple of weeks. But then the block quiz was important, so I got my act together. And so with that, I stop my screen sharing, and we take, everyone is doing then, hold on, screen share is...
Yeah, so maybe the first 30 seconds, all the students and the assistants can turn on their screen as well.
Hold on.
So I see 5, 10, 20, 25 pictures on one screen. Is there ways to have 50 pictures on the same screen? Does somebody of the students know? Or is that the limit, 25? Nobody knows a way to put more than 25 on the screen.
If you could change the resolution of your screen to a higher resolution, and then...
Anyhow, also the students, you can display now the way you want. You can remove the chat, for example, I guess. So why don't you also play around a bit and send us your pictures?
Scheider, you're still on, I see your camera. You're not a student. Are you a PhD student? But...
So there's some students don't have a camera. They wrote me that they don't have a camera.
They have to paste a picture.
Yes. So 15 minutes, no, not 15 minutes. 10 seconds, smile, also the assistants.
Also,
and
okay.
Then the assistants.
Yeah, hold on.
Okay. And then the assistants turn off their camera. Hopefully they would disappear then to the end, but
But you can also, hold on, I don't, not so fast. I want to have a nice background.
Yeah, play around. The students too, you can play around. You can
Yeah.
put your favorite cat into the picture if you want, or your favorite dog. I actually make iPhone cameras because because this is kind of nice. It's
not nice.
more personal. So I think we can leave it at that. Is it possible that all the non-students turn off their cameras for a second? But then there will be holes, I presume. But let's see. Can you reorganize so that... No, they're just holes. Yeah, but that's okay. That's okay. Henry, turn off your camera.
Hold on, I want to take a screenshot. Hold on, I do one more screenshot.
I'm sorry, but I think it will be very good memories. I hope this will be the only time in your life.
Oh, I didn't. Okay, never mind.
You will have a fully Zoom only block course. So I think we are done. Thank you very much. Fifth, Henry and
hold on, I'm leaving.
No, that's okay. But we make a 15-minute break so people have time to get a coffee and set up. Yeah. So we start these individual meetings for the talks in 15 minutes, which is 35.
When are the fish talks?
You have to figure out. Write me an email.
Okay.
Okay, have a nice break.
Ciao. Why can't I?